So welcome students. Our number is 14, so we can wait 2 more minutes, then we'll start. Okay, students. Now I think number is constant. No one is coming. No one joining further. Okay, so we have done with our module 2 also. So that is a as I mentioned, it's for the mainly purposes of detection to find the presence or absence of your bodies of interest. And so today's session we are going to for the discussion part. If any one of you have any issue in your southern blotting, western blotting, northern blotting, and your radioactive or non-radioactive jobs, what are the benefits and difficulties of any one of these two? Please raise. I'm audible to all?
Yes, sir.
Yes, sir.
So is there any issue? Is everything clear?
So the slides for the second module, they aren't on LMS.
Yeah, that I will upload.
Okay. No doubts, sir, from our side.
So that I will upload in, I think today or tomorrow. I will. Because thing is, slides are a bit segregated, so two, three different presentation they are. So I will compile according to your requirement, and then I will upload. Okay, so if no queries, so everything is fine. So then, however, this today's session dedicated for discussion, but if no question, then we can't do anything. So then we will move to the next module, module number 3. And so with introduction of the module number 3, I would like to ask you one thing. So would you like to appear for a test for module 2, or would you be happy to have a combined test for the module 2 and module 3? So should I take a test individual for your module or a separate test?
So what would be the date
So,
test for
Date
module?
will decide it today. So whenever you say that so this should be the date, we will go for that. So no suggestion, no one is responding. So it means I think you will be happy to have a single test for 2 modules. Okay. So yes, Yukta.
sir, I was suggesting that
हाँ बोलो, आवाज बीच में कट गई आपकी.
I do not agree.
आवाज कट गई
I
बीच में
don't
जो
think we should.
Sir, I think
Yes,
she
sir.
was suggesting that could we have a single test for each module?
Okay, single test for each module.
Yes, sir.
Okay, so then we have to finalize a date for it. A date, I think Monday will be.
Sir, will the test be like the previous one?
That will be decided on the same day. Is maybe objective one or a subjective part is going to be there.
So the only reason I asked was because if it is going to be like the previous test, then we could do it in any one of your periods, like any single period. But if it's going to be short answer questions, then we would probably need 2 periods to do it. That's the only reason I asked.
Vivek, your voice is means echo. Lots of echo is coming. So please speak it again. What you are saying, I'm not able to understand.
Sir, am I audible now?
Yes, now you are clear. Your voice.
No, I was just saying that if the test is going to be multiple choice questions like last time, then I'm sure we could finish them in one period. But if it's going to be a short answer question test, then just speaking for everyone. I think we would need 2 periods. So that's why I asked.
Okay, so whatever the way test is going to be, so I will ask only. I will ask question in only in that way that you will be able to finish within 20 minutes. So my questions are not going to be of a way that for that you need 2 hours or so way. So it's only the 20 minutes is going to be the time for your test. Whether it is going to be it's of multiple choice or it's going to be subjective. So not more than 20 minutes is going to be there for your exam.
Okay, sir.
Okay, so it is going to be we are going to keep it short, not very long thing. So then we can have go for test on Monday. I think 12:15 to 1:15 we are having a class. So will it be fine for Monday? It's a very small module, not a large number of topics are there.
Okay, sir.
Okay, sir.
Okay, so on Monday.
Sir, when you'll be uploading the PPTs, the meeting results.
Today or today itself.
Okay, sir.
Sir, if you are able to upload it today, then yeah, Monday sounds good.
Yeah, I'll do it today itself. Not an issue. Only thing is that 2-3 places I have to slides are there, so I will combine them and upload it. So otherwise, then you'll feel that the information is segregated and not information not complete. And I ask some question, then you say that so this thing was not in the notes, so in your slides. So don't think that questions always maybe from the slides, because sometimes happen while slide is going to be there. Just a minute. Yes, Devanshi, what do you want to say?
Sir, actually, I wasn't able to join the meeting, so I joined late.
So it's
Maybe.
we are just discussing about test. First, I asked if you have any issue in this module number 2. Then I asked that you would like to have a single test or a segregated test on module 2 and 3. Then students say that they want to have a single test for module number 2, so that scoring is going to be better. And then we will finalize the date for the test is Monday. Monday date is going to be 27th. Okay, so this is going to be your second test. And now if it is finalized, then we have to move to our next module, that is module number 3. So as you have test on Monday, so I'm not going to not tell you details about it. I will just introduce you the thing, and most of the things I already have told you, but some specific things with details and explanatory way, we'll talk about it in today's lecture, means in this module. And this module is talking about your, just a moment. And one more thing, I was talking about something coming from out of the slides. So there are books are there, and on online free e-copies of many books are also available. So one book is a simpler book, and it is very good book also for the Indian perspective. And the name of book is B.D. Singh, Biotechnology by B.D. Singh. So it contains many things, all various aspects of molecular biology, human technology, and the biotechnology. And it's not very costly also. If you would like to purchase it, you can purchase this book. It contains everything inside. But if you need detail, then you need to go for some specific book also. Then coming about the foreign authors book, the one book name is the T.A. Brown. It's, I think, a principal. Let's check what's this title of the book. The author is the T.A. Brown. So you can download it from the LibGen also. It's T.A. Brown book name, and its name is Gene Cloning and DNA Analysis. So name of the book is Gene Cloning and DNA Analysis. It's also a very nice book, and it's so nice and so well written that even a person who don't know English much, he will also be able to understand. The language is so nice of this book. So T.A. Brown. So it also contains all the things that you need for your exam, and not only just exam, for even for understanding it's also if you would like to refer it. So this is a book. So T.A. Brown, it's as foreign author is there, so it is going to be a bit costlier. So if Indian reprint is there, that may be a bit cheaper if you like to purchase it. Otherwise, this book might be available online also. So you can get its PDF. Okay, so just put, yeah, this PDF is there. So you can download it from there. And if I got a copy of it, I will also share it with you. So this is about your reading. So don't believe blindly on the slides. First thing I'm saying, don't just blindly think that whatever written in the slides. Sometime may happen that information that are present in the slides may be wrong. So if you're reading through the book and you found that there is some discrepancy in the slides, then we may discuss those points, that at what point slides are showing some discrepancy with the book. So sometime what happened, the expression is going to be different. The way book is expressed and the way slides are going to be expressed, so that we can rectify accordingly. So please read books, not just only as it is a fully baked stuff, so slide directly contain what you needs. So you think that you don't need to read books. So my humble suggestion is that if you want to prepare for higher studies, or if you want to prepare for competitive exams, please read books. So slides can never match books. Okay, so I'm sharing you the book. This is the, you can simply, it's easily available on the web. So just I downloaded it. So just I'm sharing you what that book is. Okay, so hope it is visible to you. It's Gene Cloning and DNA Analysis Introduction. It is by T.A. Brown. So it is Gene Cloning and DNA Analysis there. So it's 6th edition. So 6 is going to be older one. 2006, I think. 6. Oh, no, it's 2010. So it's not very old. So what you can get from this book. So the part of basic principle in gene cloning and DNA analysis. Why gene cloning? DNA analysis and its importance. Vector for the gene cloning, plasmid and bacteriophages. So this is the one, the module we are going to talk about. And purification of DNA from living cells. This is also the part that you need, that you want to do for a laboratory also. So it will also give you the all the your desired principles, everything. Then manipulation of purified DNA. So manipulation means that we talk about the enzymes. So what different enzymes are there? So how you can modify your DNA, how you can cut it, modify it. So then there is going to be a manipulation of the purified DNA. So you can get information from this chapter. Introduction of DNA into living cells. So this is going to be the transformation, also a practical that we are going to talk about this. And cloning vector for E. coli. So then you are specifically talking about the E. coli, what cloning vectors you are going to use if you are working with the E. coli. Then the cloning vector for eukaryotes. So if you want to transfer, if you want to clone in the eukaryotes like yeast and your animal cells or any other cells that is of eukaryotic in nature. So what type of vector you need, and how to obtain clone of specific genes. So this is a cloning, how you are going to perform the clone, how you are going to screen, then the PCR. So these I think these 9 chapters in the your part 1, they are covering your most of subjects that we need for your this course. That is the FRDD, Fundamentals of Recombinant DNA Technology. Okay, so some other things. So application of gene cloning research, sequence gene genome, then the application of gene cloning, DNA analysis. So this is a further advancement is there, how you are going to utilize it. But for I think for your this course, this 1 to 9s are more than enough. Please download it, this book from the website web. So if you need, this is a site also from where you can get it. Just I'm copying it and pasting in your chat box, so you can get it from there. So for that, I don't think I need to forward it to you. So just follow the link. At this link you can get it. So I've shared you link on the chat box. You can just, I'm going through the few chapters that are of our interest. So this is the one, why gene cloning is an. So this is the one that when I talk to you about fundamental, why are these fundamentals you needed, and how the field evolves. So this is a chromosomes, Sutton and Morgan, H. Morgan, they'll talk about the Drosophila. And in 1940, then every McCullough McCarthy, they performed the experiment, and the Hershey chase that the DNA is a genetic material. And so this is a bit history. I think we also talked about the history when we started. Then the advent gene cloning polymerase index is a PCR. Then what is gene cloning? So this is a very nicely written, you can see. So the beauty of this book is the language of the book. It's very nice and very easy and very understandable. It's an explanatory way, it's self-explanatory. And the diagram also you found in this book are very nice. So many of the diagram I also have taken from this book. So you can simply see the how these diagrams are well explained one by one and how this PCR is really going to occur. So this all are the chapter number 1. We're talking about this. So this is introduction 1 and introns, exons, and all plasmid virus chromosome bacterial causes. Then the vector and cloning. So this we are going to talk about. This is our of this module, and in this module it is going to talk about plasmid and bacterial causes. But we need some more vectors of like back and yak plasmid types. So this is we're talking about the plasmid. Why is plasmid is going to be important? So I will recommend you all to read this book. Even while I'm studying my studying, I used to study this book. Okay. So whatever my concepts are there, all my concepts are based on this book. Simply you can say. This is a very nice book. So 2 books I'm referring you. One I would suggest you to purchase because it's not going to be a bit costlier. That is your BDC. I think it's going to cost you around, I think, not more than 500 rupees. Okay, so this is the BDC. This is the book. This is the Expanding the Horizon is a book. It's available on the Amazon. See, it's just for 80 rupees. So it's very nice book. And anytime you would like to just quickly see what this really biotechnology is and what different biotechnological aspect you need it. So refer this book. Read this book. And even if you would like to appear for many of the exam, it covers all of the things. Okay, so that's why you need not to at your level. But if you're going for higher study, for masters and so on, then definitely you will be required some other books also. But for the undergrad level, this book is you can say it's simply it's a Bible, you can say, for you, but not for all. Okay, so these are the books that you should read and study. I don't think this BDC is going to be available online. If PDF of it is available, then you can take that also. But I think reading from book is better than studying on your mobile or on your laptop or reading, because whenever you are reading on your book, you will understand well and you will grasp well. If you are studying using mobile and on the scrolling the slides, just you will remain in the scrolling. Okay, so your mind is, I don't think it is going to be focused much. So either take to prints or read by books. Okay, so where we were, so back to our module. Okay, so this is your module number 3. So this module number 3, the title of this module is the cloning vectors, and its weightage is 25%. So this means 25% by marks and 25% by lectures we have to spend over it. So it means out of 28 lectures, 25% is going to be 7. So 7 lectures we are going to spend on this cloning vector. So we are going to talk in detail way. So your cloning vectors, what type of cloning vectors we are going to deal in it? One is based on the plasmids. So as I have shown you in that book also, that T.A. Brown, that is having the cloning vector that is the plasmid and the bacteriophages. And the second is here also you are having bacteriophages, that's the lambda and the M3, two bacteriophages are there. And third is the high capacity cloning vectors. So you need high capacity means high, you want to load more, then you need to go for the yak and back. Then the fourth type is of the vector is also there, that's called the expression vector. So now your job is not just cloning. Cloning means making a multiple copy, but you need a product also. So for that, some extra, some additional component in your vector is also being required. Some extra property your vector must have so that you can express, you can uh, show out your gene that you are cloning. So let's come to the term expression vector. So these are the components of your model number 3. So that we are going to discuss in it. Okay, so just the introductory one, as I mentioned you, so I won't consume long, means I won't, I would not like to try to confuse you because your mind might be a bit focused on your module number 2 because you have to give a test on Monday. So you don't want to be extra burden your mind because if I take long lecture of this topic, then you have to read it all again so that you can grasp it with well. So this is the thing. Okay, so here the term we are saying vector for gene cloning. So first, simply think, but as I want to ask you, as all of you, we have studied, I think many of you also have it, and all of you have uploaded it on the MS Team also for part of your laboratory. So first thing is we should understand what this gene cloning mean. What is cloning? Because what is cloning? Simply cloning. What do you mean by cloning? Because it's introductory we have done in our last module itself. What do you mean by cloning?
So cloning is the process of making a copy that is genetically and morphologically identical to the original.
So yes, yes, Manashwini. So as Manashwini mentioned that cloning means replica, making replica. So replica means whatever things you are saying was present in one is also present here. So when you are preparing a replica means it should behave like in a replica and it keep on producing the replica. So we are going to, when we are seeing the cloning, in this cloning we simply means we are going to generate a system whenever we need a copy like photostat machine. So when you put a paper within the photostat, so just you need to press a button and photocopier just start copying it. So whatever was there, it is copying exactly what is there. And when you need if you need a black and white copy or if you need a colored copy, so what type of the replica you need depending upon your requirement, you put feed your information there, or you can get it. So here also the thing is there. So when you want to generate gene cloning, so gene cloning means you are want to prepare the replica of a gene. Your gene, one copy is there, you want to make multiple copies, 2, 3, 4, so on. Multiple copies of same gene. So when we are making multiple copy of a gene, and in first lecture itself, we described that two ways we are going to do while preparing this copying. And in these two ways, we also mentioned that one way is the cloning, and second way is a PCR. So in PCR and cloning, the way you are copying it's going to be different, but end result is going to be the copying. So simply thing is that you want to gene, as we all know that by the definition we mentioned that it is going to be a sequence of DNA which is going to code for a function or code for a phenotype or code for protein, you can say. But in true sense, when we are talking about the gene, that it simply means a DNA sequence. So what DNA sequence, what you are proposing that should be there when you are going to call it a gene, but it is a DNA sequence. So this DNA sequence is a DNA. So to display the feature of a gene, the feature of a DNA, a displaying agent also that we need, it should be a DNA only. So the vector, that is simply term when we define this, what this vector is going to be, we call that this vector literally mean a vehicle which we are going to use for the transport. So when we are using some gene cloning or the molecular cloning, in which if we are using a vector to clone it, so that vector should have certain properties, the vector should have certain features, so that it can act as some vector. So like as I mentioned, when we are calling our bicycle is a vector, it has certain property in that, and that property is that it has space to carry you. And not only the space it can carry you, it can move also, it can serve as a shuttle. It can take you from one location to another location. So, and one more thing with the property should be, it should be always available. Like it should be like metro. And let's say it's like metro means whenever you feel that you have to go, just visit the metro station. You will find a metro either it is on the platform or coming in 1 or 2 minutes. So the replicative one, so it is you can replicate it also. These are the certain property that if your DNA molecule is going to have, then we are going to call it as a vector. So DNA molecule need to display several features to be able to act as a vector for gene cloning. So what are those vectors going to be? So as I mentioned, first factor that you need it should be able to replicate. Because when we said that the cloning means you want to maintain the gene. You want to make that gene should always be there. So for that, and not just it is going to be there, it is also going to be uh, uh, transferable to next generation. Because a population of cell, we are if we talk about a biological cell, if we talk about, so biological cells replicate. What are type of cells we are talking about? Whether it's a bacterial cell, animal, plant cell, animal cell. So they replicate. So when they replicate, their nucleus, their DNA that replicate, it forms another copy of it. And when you form another copy of it, half of this copy is going to one cell, and second half is going to the another cell. So both cells become alike, and DNA, the amount of DNA or the quantity of the DNA is retained. So this property should be there within your vector also. That means it can replicate. Because what happens if your vector is not replicating, but cell is meant to replicate. Okay. So if cell is going to replicate, then what is going to happen? Then the next generation is not going to have the vector DNA within it. So if the next generation is not having the vector DNA within it, then it's not going to be vector. It was just a DNA, and that DNA you transferred, and it stayed in the cell for a certain time, and when it replicates, it loses off. So then that is not going to help you anyway, because once one molecule that you have added to the cell is remaining that light. And not just it is not just one molecule, also when as the cell dies, the DNA also dies. It also got degraded. So that means if your cells are replicated, and with the replication of your cell, your DNA molecule, your plasmid that you are talking about, your vector that you are saying, will also replicate. So this is a must be able to replicate within the host cell. Then, as I mentioned, we are talking about every replication means it is making copies. When you go from one cell to another cell, it has to make copies. So then the question arises: how many copies of your vector you need? When we are saying the copies of vector, it simply means we are talking about the copies of our gene that this vector is going to carry. So in that case, if we ask that how many copies of the gene should be there, how many times it should replicate? So then, सिंपल सी बात हम कहते हैं, ज्यादा किसको अच्छा नहीं लगता? Everyone likes more, and everyone wants more. So in this case, if we are asking for multiple copies, we want that if within a cell, if these copies are relatively large in number, in that case, you don't need very high number of cells. This simply means if your DNA replicating 100 times within your cell, so then if you take out your DNA from the cell, then you are going to get 100 copies. But if your plasmid DNA is replicating once only, then what is going to happen? Then if you take out the plasmid DNA from the cell, then what are you going to get? You are going to get just 2 copies. Or that second copy that you are referring might be ready to divide, so it will move to the second cells. Okay. Just wait a moment. I'm having some water, so I will join in just 1 minute. Yes, then simply you can say that it should be having the numerous copy. So that because our aim is to have multiple copies. Cloning simply means having copies of it. So if within a cell, within a host, if you are having a numerous copy, then you don't need a large number of the cell to have desired copy of your gene or desired copy of your DNA. So this is the second property. Then coming to the pass it to the daughter cells. So you may say that it is replicating and now it is going to have multiple copy, but it is not passing to the daughter cells. Then the same issue is going to come as cell die. Your vector also die with the cells. So we want to make it like that. It is going to sustain till eternity. So till the end of the arc, it is going to stay. So that can happen only if this vector have an ability to pass through the daughter cell. Means when it is replicating, that's fine. But when cell divide at that time, that as you know that in case of mitosis, then the whatever is arranged on the metaphysic plate, that is going to migrate toward the poles and will be part of your cells. Similarly, your mitochondria also they also replicate and they also move to the respective cells when cell divides. So similar property should be there with this vector also, that it must be passed to the daughter cells. Daughter cells means those cells are going to form after cell division. Okay. So then again coming to another property that your cloning vector should have, that is it needs to be relatively small. So relatively small, that if you remember that we talked about, small means if it is going to be small itself, like you know that uh, uh, about your cars, like Vivek might be having a better experience for it. So those which are having racing cars and racing bikes and other types of the cars that we are saying, which having the high horsepower engine, or in Indian context we are saying a high CC engine. What is happened with that? Or simply one more example, let's say, like you are having a car and in which is a CNG fitted. So when you are putting CNG cylinder itself is a very high, very large weight. So you can't put a very large capacity CNG cylinders. What is going to happen? This car's own weight is going to become very high. Then what is going to end this with? It is going to end with a very low efficiency of your car, which your car is going to take you just few kilometers in a liter. So then similarly going to upper in case of your vector also. So if the size of your vector is going to be very large, then in that case what is going to happen? It cannot carry the amount of the load that you need, and second thing, it is itself so large that there is going to be no entry zone for this large particle. So like no entry zone, you don't allow heavy vehicles. So similar type of thing is there in case of cloning vectors. So your host behave like a no entry zone, and it won't allow the heavy loads to enter within it. Because if heavy vehicles are entering in your in your streets, then your heavy vehicles are going to destroy your roads. A similar thing may occur in case of your DNA vector also. So it must not be very large size, should be relatively small, and ideally less than 10 kilobase pair of the size. Then next property that it should be there, large molecules tend to break down during purification. So if the molecule is going to be large, like in the case of DNA, when you purify DNA, it is very difficult to have intact DNA. So what happens? DNA have ability to shear it down. So this, if it is going to be very large in size, then might also possible that while you extract it, while you take out the DNA, while you take out the vector, then it may also degrade. So it got degraded, it got broken down in your extraction process, then your job is over. So it's not going to be beneficial for you. So large molecules tend to break down during purification. That's why also we need to keep it small in size. And another thing, if it is going to be large, then further become difficult to manipulate. So manipulate means change you want to do in your vector is not going to be possible for you. Okay, so two kinds of DNA molecule are able to satisfy what are the criteria we have mentioned above. That these criteria are there, and they are self-replicative. They replicate multiple times in a cell. They retain themselves, and when cell is dividing, they are also migrating to the next cell type. So this is the thing. There's a two things that we are mentioning, and we are going to utilize these two properties only. So what are other types of vector that we are going to call about? They all are going to be based on these two, or maybe minor variation of these two. Okay. So this is just introductory one for your learning vectors, and in detail we will do in our next week. And with this, I would like to conclude my speech, and I would like to ask you to please turn on your camera when I call your name. Okay, so Adish,
Yes, sir.
Ayushi, Ayushi, Adish, turn on your camera. Ayushi,
Is it visible?
yes. Demetrius,
Present, sir.
Devanshi,
Present, sir.
Ishita,
Present, sir.
Janvi, Janvi. Your voice is not audible, Janvi. Janvi, you told that there was an issue in your attendance in previous class. You are not audible. Your mic is not working, I think. No. Okay, you can type it. Next is Kshitij. Kshitij. Manaswini.
Present, sir.
Namrita.
Present, sir.
Nehal.
Present, sir.
Rupal.
Present, sir.
Saiba.
Present, sir.
Sanjana.
Present, sir.
Shakti.
Present, sir.
Shivani.
Present, sir.
Tanvi. Vivek. Yukta.
Present, sir.
Yuvraj.
sir.
Present, sir.
Present, sir.
Okay, so Tridev. Three students are absent today. So can you name them? Who are these three? So Amazon is not opening. So
Sir, I think the Dev is not in the call.
Yes, sir.
one is Tridev और कौन है भाई?
Sir, I think that's it.
नहीं, three students are absent. I'm missing Atoiba.
And Kshitij, sir.
Yes, sir.
Kshitij
Kshitij
was
is on.
present. So Kshitij is coming and going.
Okay.
Atoiba and Tridev. These two. One more is there.
Sir, Nandini,
Nandini also. Something she was there. I think she left. I think.
Sir, Nandini is having network issues.
So Atoiba and Tridev.
Yes, sir.
Okay, so thank you all. Stay safe, stay happy. So Janvi, you are. I checked. You are present. You are in my attendance register. You are present in all of your two classes. Check it again at your end. And if you found the issue persistent, show me in which class you are absent. Okay. So thank you all. Stay safe, stay happy.
Thank you, sir.
Thank you, sir.
Thank you, sir.